Howdy, folks. Thanks for checking in to Mr. Ulrich's Land of Biology. I, of course, am Mr. Ulrich. And in this video, we get to start talking about sex. Yep, that's right. That's what we're going to talk about. We've already compared asexual reproduction to sexual reproduction and talked about how it's not the number of parents, as many people would think, that makes the difference between sexual reproduction and asexual reproduction. Where in asexual reproduction, you start with one cell and end up with two identical cells. In sexual reproduction, you have two cells and they fuse to become a new cell. And in doing so, if there are two parents, then it's going to share the characteristics, some of the characteristics of each parent, because each cell is going to contribute half the genetic information. Now, what we're going to look into is the type of cell division that is going to make those cells that are going to come together and fuse, and talk about how you can get genetically different gametes, even if we're getting them from a hermaphrodite, a single parent. Since mitosis is the asexual version, you end up with identical cells. Mitosis is not going to be able to make these cells that are going to fuse together. We need a different type of cell division. When we think about the types of organisms that engage in sexual reproduction, it's not unusual for us to associate it with animals because all members of the animal kingdom engage in sexual reproduction, even the vertebrates, right, like these guys, and even invertebrates like insects and crustaceans all reproduce through the fusion of gametes. Though, as we have seen, some animals are capable of also reproducing through asexual means. By fragmentation and other ways. Sexual reproduction is not the only providence of animals, however. Plants also are rather sexy. The flowers are the sex organs of the plant. Some flowers are complete in that they have both the male and the female reproductive organs, although some flowers are incomplete, where you'll have, like this bladder campion here, you'll have male flowers and female flowers different from one another. Regardless of whether we're talking about plants or animals, those specialized cells that fuse to form a new individual are called gametes. The male gamete is the sperm produced in the male gonad. No, gonad is not a bad word. It's a technical term. You can use that. The male gonad of the testes. Now, testicles are testes. They're just kept outside of the body, well, in a scrotum. The term gonad is also not reserved for males. There are female gonads, that is the ovaries, or those are the ovaries, where ova are produced, the technical term singular ovum. The old joke I use, of course, is next time you go to the diner, you can order your ova over easy. Regardless of whether we're talking about plant or animal cells, we can categorize cells, as we're thinking about gametes, as whether they are gametes or not. And if they're not gametes, then they are what we call somatic cells or body cells. Now, these are all the cells of the body. These are the ones that, well, they originated from that first cell that came from those two gametes coming together. What that means is, since each parent donates genes for each trait, that means that your genes for traits come in pairs. All right, most sexual reproducing organisms are diploid, meaning that their genes for traits come in pairs, one from mom and one from dad. And since all of your genes are arranged on chromosomes, that means your chromosomes come in pairs. Right, one from mom and one from dad, right? And so the fact that they come in pairs is reflected in this number 2n. That term n talks about the number of chromosomes that are in a set, because not all organisms that are sexual are diploid. Some of them are tetraploid, like trout or strawberries, which are octoploid, so 4n or 8n. Now, the other cells then, the gametes that we've been talking about, those sex cells, the sperm and egg cells, since we're forming from the fusion of those cells a 2n number of cells, we have 23 pairs of chromosomes. So 2 times 23, we have 46 chromosomes. That means I have to make 46 chromosomes from the fusion of two cells. All right, that means I'm going to have 23 and 23 coming together. So each cell has one of each pair of chromosomes. So since they have half the number of chromosomes, they are haploid, right? Half or half. Di for two, half or half. We could also call them monoploid, since we are diploid organisms, right? Even so, they have just one chromosome in each set, and so their symbol would be N. Fertilization then is the fusion of a sperm and an egg cell forming that first somatic cell, the zygote. So 23 chromosomes in the haploid egg fused with the 23 chromosomes in the haploid sperm cell, forming a diploid 46 chromosome or 2N zygote. That zygote is then going to divide, mitosis, blah, 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 and down the line, making lots and lots and lots of other cells that have 46 chromosomes. They're 2N. This concept of diploidy, that chromosomes occur in pairs and that you get one from mom and one from dad, becomes really important when we start getting into genetics and looking at patterns of inheritance. This is what we see diploid in most sexual organisms, though, like I said, there are variations. There are tetraploid trout and octoploid strawberries, to name a couple. Regardless, even though most organisms are diploid, each species has their own characteristic chromosome number. Now, it's not like each one has one that they own. We share 46 chromosomes with mice. That's why a whole lot of research is done on mice. Now, we have a lot of genetic similarities with mice, but our closest genetic relative is the bonobo, the pygmy chimpanzee. And we share a lot more genes in common with pygmy chimps, yet they have 48 chromosomes where we have 46. So that very similar DNA is arranged a little bit differently, that's all. When we start thinking about diploid pairs of chromosomes, it's not any just random pair of the 46 chromosomes that makes this diploid pair. The two chromosomes that come together to form a structure called the tetrad, which I'll show you in a minute, are actually called homologous chromosomes. The prefix homo for same and logi. Remember when we learned about biology as the study of life? Everybody says logi is the study of. Well, it's really like the information about. When you study something, you learn the information. So that kind of makes sense. So what does this mean, homologous, same information chromosomes? Well, you get one of each pair from mom and dad. It doesn't matter whether it's mom's or dad's. They're about the same size and shape because they carry on each one the same traits. So they carry genes that encode for characteristics of the same traits. They might be different characteristics, but they are of the same trait. This means that if I look at one location on a chromosome, a locus, say we use the same term as in math. If I look at one locus of one chromosome, and I look at the same locus, the same location, the same spot on the corresponding homologue, right, that location is going to code for the same trait. Though it might be a different gene. Here, what do I mean? So when we start looking at homologous pairs of chromosomes, right, up here is one location, right, and here is a gene that codes for eye color. Maybe this one comes from dad and codes for blue eyes. All right, over here if this is dad's, right, let's remember this, dad. Okay, if this is dad's, right, over here on mom's chromosome, mom's contribution to the homologous pair of chromosomes, at the same locus, I'm also going to find a gene for eye color. But it might not be the same gene. Maybe mom's donating a brown eye allele, right? It's called a gene that codes for brown-eyedness, right? And that's the case with all of the different genes at all of the different loci, all along here, right? Right. Maybe this one also has maybe dad's also got blonde hair, right? Right. And mom's got brown hair, right? So these pairs of homologous chromosomes, though they carry the same traits, might carry different characteristics of those traits. Now, it's also important for us to recognize if right here is blue-eyed on the same spot, right? Since these two chromatids are the result of replication, right, is going to be the same gene, the same trait, right? It's going to be blue eyes both here and over here on the homologue, right? So we call these what we call sister chromatids, right? Because they are the result of replication. So here would be brown eyes as well on both of these. Right, and there's 4 chromatids. That's why we use the term tetrad, right? Because of those 4 chromatids that make up a homologous pair of, in this case, double chromosomes. So this means then that we have 23 pairs of homologous chromosomes. Now this it's going to get a little different, okay? Once we start looking at what are called the autosomes, which are like the body chromosomes, compared to the last pair, which are called the sex chromosomes, we're going to find that males, most typically as determined at birth, don't have homologous sex chromosomes. So saying that all humans have 23 pairs of homologous chromosomes isn't always accurate. A little less than half of us have 22 pairs of homologous chromosomes and 1 pair of non-homologous chromosomes. But we'll get into that here. Here's a photograph of actual chromosomes. This is called a karyotype, and I'll show you how we make these in a bit. But they're arranged in pairs. So here's one and here's the other of a homologous pair of chromosomes. This isn't one double chromosome. Each one of these, here's one doubled chromosome, and here's another doubled chromosome. I know they don't look doubled, so the 2 chromatids are like smushed right up against one another, which they will do, and they'll do when they form tetrads, which we'll get to later on. So like I said, these first 1 through 22, they arrange them by size, are called the autosomes or body chromosomes, and the last 2 um, are the sex chromosomes. Now, sex determination gets when I teach sex determination, I focus on the typical presentation of sex. I'm fully aware of the difference between sex and gender. I try to be as sensitive as I can to transsexual, intersex, all of those things. Sex determination is really, really interesting and not always as straightforward as it's made out to seem. I'm always trying to do my best. So if I make an error, feel free to send me an email or DM me and correct me. I'm just trying to do the best I can. So with that said, here we go, talking about sex chromosomes and sex determination. Most typically, when those sex chromosomes are homologous, when there are 2 what we call X chromosomes, I know they all look like X's, different sizes. When we're talking about the sex chromosomes, right, when we talk about 2 of them looking like X's, right, that most typically presents as female. The male sex chromosomes, like I said, are non-homologous because when I look here, over here, I might find a gene for a clotting factor. I'll find it here, I'll find it here, I'll find it here. Heck, I'll find it here and I'll find it here because it's on an X chromosome. Right now, when I look at the Y chromosome, that's there. Well, there isn't a locus up here now, is there? Because that Y chromosome is a little bitty thing, and there isn't much on the sex chromosomes. Now, it might be tempting to think about all body traits being on the autosomes and all of the genes for peepees and hoo-ha's being on the X and Y chromosomes. They are not. Okay, I just identified a gene that's actually on an X chromosome, which is a gene for a clotting factor. There are a whole host of genes on these chromatids, on the sex chromatids, that have absolutely nothing to do with sex. What there is, right, there is one location on the Y chromosome. It's a group of genes that code for proteins that act as switches that turn on other genes that code for peepees and hoo-ha's for all those parts, really code for the other proteins that will influence the development of gonads and secondary sex characteristics. It's called the SRY, right? The sex determining region of the Y chromosome. Interesting, sometimes this SRY, as other parts of chromosomes can break off and join other chromosomes, does happen, right? It doesn't happen all the time, but it can happen, and it can be, a lot of times people don't even know that it's happened. Right, so sometimes this a piece of the X chromosome can break off and be added to any other chromosome, even an autosome, right? And the person might have 2 X chromosomes, but somewhere else have a little piece of the SRY, and that person will most often present at birth as a male. Right, even though genetically they're XX and would be determined through a karyotype as a female, most likely. Right? The males are said to be XY, as determined at birth again. So here's a karyotype of a typical female at birth, all right, being XX. Again, we could have a piece of the Y chromosome, the SRY, stuck on to another one somewhere, and that could happen. It is rare. I don't want to say it like it happens every day. It doesn't happen every day. Although the most common mutation is a lack of an X chromosome. Right, missing an X chromosome. It's a version of what's called aneuploidy, and it's survivable. That's why it's so common, because half of the population only has a single X chromosome. So having only one X chromosome is totally, totally normal. And so people with, it's called Klinefelter syndrome, people with Klinefelter syndrome, though they present as female, only have one X chromosome. They present as female because they're lacking a Y chromosome with an SRY. Now, when it starts to make those cells that are going to carry each group of those one set of homologs, right, to come together and fuse and rejoin that number of chromosomes, right, we need a form of cell division that is going to form cells that only have, well, for us, 23 chromosomes in each one of each pair. All right, and that type of cell division is called meiosis. It's very similar to mitosis, but what happens is we end up reducing the number of chromosomes, or reducing the amount of genetic material. So it is often referred to as reduction division. Most organisms, sexually reproducing organisms, are going to go from a diploid cell down to a haploid cell. So I can take haploid plus haploid equals diploid. For some reason, the Regents used to love this. If we made cells, gametes, mitotically, then we'd end up doubling the number of chromosomes each generation, and certainly that would not work. What's cool is that we don't have, cells haven't like engineered a whole nother set, a whole different way of moving chromosomes around and dividing cells. It actually uses the exact same cellular machinery that we saw in mitosis. So the setup and the execution is very, 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 very similar, with a couple key differences. For one, we're going to kind of stack 2 rounds of mitosis on top of one another, and we're not going to duplicate the chromosomes, because remember, we're trying to halve the number of chromosomes. So this is going to use the same phases, like I said, the same machinery that mitosis uses, right? Except we're going to reduce that number by half, right? So we start out with prophase, just like we saw before, right? Metaphase, where the chromosomes line up, anaphase, where they start to divide, right? And telophase, where they finally pinch off and you get your resulting cells. That's going to happen in meiosis 1 as well. I'm going to blast through all the phases and what happens in pretty rapid succession. Feel free to hit pause so that you can write down some notes, go back and watch it again. If you're, well, even if you're doing this on an end puzzle, you can go back and watch it again. You just can't skip forward. So prophase 1, like I said, it's going to look just very, very similar to prophase in mitosis. It's really long. We got to move the centrioles. We got to build the spindle fibers. We have to condense the chromatin into chromosomes, right? All of the same stuff. One of the big differences is now, remember, we got to focus on these homologous pairs of chromosomes. So where individual chromosomes would be attaching to the spindle fibers, right? In our cells be 46 individual chromosomes. Now, we're putting those homologous pairs together into those tetrads, and we're connecting those tetrads. So now there's only going to be 23 structures that are being attached to the spindle fibers. This formation of tetrads is called synapsis, a term that you're never going to be quizzed on, but it's kind of cool to know, right? That we're bringing together these non-sister chromatids that are carrying traits for the, or genes for the same traits, though they might be different characteristics of those traits. So here's a question for you. If I got a cell, diploid cell, typically has 20 chromosomes, right? At the end of meiosis, how many chromosomes are we going to have? Pause. Oh, wait. Of course, we're going to have 10 chromosomes. If we start out diploid 20, right? Haploid is half-loid, so half of the 20 chromosomes is going to be 10 chromosomes. Here's another question for you. Now I got a cell that's got 40 chromatids at the beginning of meiosis, right? Now at the end of total meiosis, not just meiosis 1, at the end of total meiosis, how many chromosomes is each cell going to have? This is a hard one. Oh, wait. You can pause it. All right. Well, it's important to know for this one that when you go into meiosis, at the beginning of meiosis, all the chromosomes are doubled. So that 40 chromatids actually equals 20 chromosomes, right? So it's actually the same question as the previous. The answer, of course, is 10 chromosomes. All right. Now, it's important to note that at the end of meiosis, just like at the end of mitosis, I'm not going to have double chromosomes. I'm going to have single chromosomes. And that's actually an important idea, right? That you're going to have single chromosomes at the end of active mitosis, meiosis, so that it can then go into interphase, right? Or in the case of gametes, fertilization, and then go into some interphase so that we can double that DNA and get ready for mitosis. All right, so if we start out with 40 chromatids arranged in double chromosomes, I'm going to end up with gametes that have 10 single chromosomes, which is kind of like 10 chromatids. So in prophase, what it's going to look like, here's a cell that has 4 chromosomes in 2 pairs. All right, just like we saw before, except the difference is that now they're not lining up individually, they're lining up in tetrads. In metaphase, we're going to see those tetrads line up across the middle of the cell itself. There is a variety of ways that these homologous pairs can arrange, whether the maternal or paternal, because remember those genes might be different even though they code for the same trait. So whether the paternal is on one side or the other side makes a difference, because now it matters which gamete it ends up in. All right, metaphase, of course, just like in metaphase, the shortest point when they line up across the equator. Anaphase, they separate, but now it's important again to recognize that we're separating a homologous pair of chromosomes, a tetrad, right? So now, as we go through anaphase, right, and then into telophase, right, our resulting cells are going to be doubled, right? And so they're not really ready to fuse yet, right? Because once they fuse, they have to replicate. These are already replicated. So we need another round of mitosis, really meiosis 2, to separate these double chromatids, or double chromosomes, into individual chromosomes. And that's what the next round is going to be. We're going to kind of go into interphase real briefly if you're a male. In females, interphase actually is very long for some cells. We'll talk more about this when we get into the differences between spermatogenesis and oogenesis. But generally speaking, we talk about interphase being suspended because there is no DNA replication, whether you're talking about boys or girls. Between meiosis 1 and meiosis 2, there's no DNA replication. Remember, we're trying to have the number of chromosomes. And by the end of meiosis 1, we have halved the number of chromosomes. We started out with 46 chromosomes, and now after meiosis 1, there are 23 double chromosomes. Doesn't matter that they're doubled, that's still individual chromosomes. So we've halved the number already. So here we go. We go through prophase 2. We're going to line up. Now it's going to look just like mitosis, except because we don't have homologous pairs, right? So it's going to look just like mitosis, except there's half the number of double chromosomes to line up on the equatorial plate. Again, there's a variety on the way that these can line up, maternal versus paternal. And since maternal and paternal can be different, when we go through anaphase, right, separating the chromatids, and then finally telophase, reforming those nuclei again, and cytokinesis, we end up with 4 haploid cells, right, those sperm or egg cells. But it's important for us to recognize that those 4 cells are genetically different from one another. Right, so here's a maternal and a paternal, here's 2 paternals, here's 2 maternals, here's a paternal and a maternal, but the other way around. So each one of these gametes is genetically distinct, right? And if we were talking about our friend the tapeworm, hermaphrodite, who makes both the sperm and the egg cells, right? If the parent cell looked like these guys, more like these guys, right? We could end up with 2 gametes of this genotype fusing, right? And then we would essentially lose, in this case, the father's contribution to those to the zygote. Now that doesn't actually happen because there's crossing over, which we're going to get to later on. But what you can see is the degree of genetic variation that there is amongst the gametes. Right, and that you're going to have cells that are genetically distinct from their parents through meiosis. Speaking of gametogenesis, when it's meiosis in testes, it makes sperm and it's called spermatogenesis. When it's in the ova, it's called oogenesis or oogenesis. Yes, you are supposed to pronounce each one of those O's separately. So when you go to the place with all the animals, it's called the zoo. In spermatogenesis, we start out with one primary spermatocyte or primary sex cell that goes through meiosis 1, and then those 2 go through meiosis 2, and I end up with 4 sperm, each one with their haploid number of chromosomes. Right, this begins at about puberty and continues throughout the entire male's life. All right, a little different from this is oogenesis. In oogenesis, though there are 4 cells that are produced, cytokinesis is unequal. And so from each primary oocyte, we only end up with 1 actual egg. The other 3 cells that are produced are called polar bodies and are reabsorbed and are not used. They don't contribute. Right. So all of the cytoplasm ends up going into the big, huge, going to be an egg cell. So they get all of the mitochondria and all that kind of stuff that they're going to need. The sperm doesn't provide any of that stuff. All the sperm provides is 1/2 of the nucleus. I told you I would talk about how we make a karyotype and got those groovy pictures. This is done through a process called amniocentesis, which isn't done as often. Procedure, you like that? Which isn't as done as often. My wife and I had a high-risk pregnancy because we were kind of old, and we didn't have an amnio done. The way it works is when it is done is they use a big needle, big scary needle, withdraw a little bit of amniotic fluid from surrounding the placenta, collect those cells, culture them, and then find cells that are going through mitosis and take a picture of them. We can arrange that picture of cells, right, cut and paste and arrange them into a picture and make sure that there's the right number of them. We can even tell if it is most likely going to present as a boy or a girl. This, of course, would be a boy, as most typically presented at birth. Right. We can also find out if there are versions of polyploidy, right, which is when you have an extra number of chromosomes. This is actually called trisomy, right, because we're talking about 1 pair. I guess polyploidy is when you have a whole set of extra chromosomes, right? So instead of being diploid, you're triploid, right? So instead of there being 2 first chromosomes, there's 3 first chromosomes, and then instead of there being 2 second chromosomes, there's 3 second chromosomes. So you have an extra one of each one of these. Individuals who are triploid, right, tend to be sterile because they can't make tetrads, right? Individuals who have trisomy 21, like this individual does, also tend to be sterile because they can't make tetrads very well. Um, and this does cause some pretty major developmental problems having an extra chromosome. Most versions of trisomy are not survivable. Like I said, the most common version of aneuploidy or of having the wrong number of chromosomes is Klinefelter syndrome, having a single X chromosome. Oh my, that is a far more than I probably should have covered today, but whatever. We have a lot of ground we got to cover. I hope you all have a lovely weekend. Enjoy the wellness day. Enjoy the weekend, and I'll see everybody on Tuesday.